Good afternoon. Welcome to today's class. So today we are going to a new chapter, that is microbes in human welfare. So what does microbes refers to? Microbes refers to microorganisms. So microorganism consists of what all organisms? Can you name it? Microorganisms, a group of organisms which consist or which includes bacteria, fungi, algae, virus, etc. Actually, in the last class, we learned about the human health and diseases where we learned about pathogenic disorders. It is pathogenic in the sense the diseases caused by pathogens. Okay, so pathogens included the microorganisms there also. Now, in this chapter, we are learning about the microbes in human welfare. That means which are useful for human beings. That means which benefit human activities and welfare. So yes, microbes play an important role in a variety of processes that help in fields like health, agriculture, industry, industrial application, that is about industrial microbiology, as well as environmental sustainability. So environment. So coming to health, which is related to the medicine. So we know the discovery of antibiotics was a life-changing or a big change to the medicinal or the health industry, where it was like a change or it was like a blessing for curing many kinds of diseases. So, for example, the penicillin, which are derived from the fungi and bacteria, which help to fight the bacterial infections. And also we can include vaccines, which certain microbes or their components are used to create vaccines. We learned it in the last chapter. So these vaccines are also made up of the antigens, or we can say the dead or inactivated microorganisms which are injected to our body. So yeah, these also help in preventing various infectious diseases. For example, the polio vaccine, which are made using the weakened polio virus strain. Okay, now moving to the next category, that is coming to the agriculture. First thing what comes in our mind is the nitrogen fixation, isn't it? Certain bacteria like rhizobium, acetobacter are involved in fixing atmospheric nitrogen into the soil. And it also includes a biological pest control. So it's like some microbes help in controlling the agricultural pest, for example the bt, bacillus thuringiensis. And it will also include the composting as well as. And coming to industrial applications, the first term that comes to our mind is fermentation. Okay, so we have learned a lot about the term fermentation. So what is fermentation? Like certain microorganisms use this process for producing, like food materials like alcoholic beverages, like wine, beer and bread. And what they do is that they convert the sugar into alcohol and carbon dioxide. Then comes biofuel production. It is also useful in food industry, biodegradable plastics, etc. Moving to environment, they are like helping in biodegradation, bioremediation. That is in like particularly in waste management. Now moving to environmental sustainability, we use microbes in waste water treatment, carbon cycle, the soil fertility, etc. And one more branch should be included, that is biotechnology. So yeah, we use microorganisms in gene cloning, and also we use for like some production of bioproducts as well. So we can say that the microbes are essential to human welfare in many ways. They contribute to, as I mentioned, health, agriculture, industry, waste management, industrial industrial applications, environmental sustainability. So their ability to decompose the organic material, to fix the nitrogen, to produce medicines, and also to contribute for the energy production makes them indispensable to modern society. So whenever this chapter is coming to your mind, you should collect all these terms, the organisms, and they all together as decomposers, as well as they are able to produce some byproducts, for example, the antibiotics, and they are also used as vaccines. And in agriculture, they are able to control pests. So many products are produced from bacteria, etc., etc. So we'll go into all these topics in detail in the coming slides. So again, welcome to this class. So we are moving to the chapter Microbes in Human Welfare. So as I discussed, microbes are diverse, which includes protozoa, bacteria, fungi, and microscopic plants, viruses, viroids, and also prions, that is proteinaceous infectious agents. So this term is very important in NEET exam. What are prions? So prions are the proteinaceous infectious agents. So you have to remember this term. Now, the microbes like bacteria and fungi can be grown in nutrient medium. In this diagram, you could see this is a nutrient medium to form colonies. These are colonies and can be seen in naked eyes. So what is this nutrient medium? So we know that as human we need energy, and for the energy we consume food. Okay, so we take carbohydrates, proteins, amino acids, lipids, all kind of vitamin, minerals, water, everything from the surrounding environment, and we absorb it, and we take it for our energy utilization as well as for our metabolism. For our daily routine, we need all these nutrients, all the elements present on earth. So now they are also living organisms. Microbes also need energy. They also require nutrients. So we will be growing these bacteria or microorganisms in vitro condition. That is not in vivo. We are talking about in vitro. That means in outside the organism. That means outside our, that is like in a laboratory we are cultivating them. So we are taking a petri plate where we'll be adding nutrient media. So this nutrient media will be containing, like for example, they'll be having a carbohydrate in that media. For example, it might be glucose, it might be lactose, or and it can also contain so much proteins. Peptide growth, like we'll be adding so many kind of nutrients into it. Agar-agar is added. Okay, so we add the nutrients whatever are needed for the growth of bacteria, fungi, or whatever the microbes are. And some microorganisms cause diseases and some are useful for human being. In the last chapter, we learned about the diseases caused by some microorganisms. And here in this class, we are learning about the advantages or the applications of microbes in the human life. So now microbes are present everywhere. So like right now, wherever you are staying, you might use a microscope. You see at least 20% of microorganisms around you. At least 1% you can find around you. Okay. Even in your body, inside your body, you have the normal flora which are needed for your growth, not growth, for your metabolism, for your sustainability, whatever it is, like the normal mechanisms to for the stomach, also the intestine, also you have a normal flora. Okay, so the microbes are present everywhere. For example, in the thermal vents of the geyser, its temperature is about 1000 degrees, 100 degrees Celsius. There also we have the microorganisms. And deep in the soil, under the snow, diverse protozoa, bacteria, fungi, virus, viroids are present. Now, one among the applications of these organisms are they produce antibiotics. And one among the disadvantage or the harmful effect of these organisms are they cause diseases. Now, moving to microbes in household products. So common example is the production of curd from milk. So we know that, right? Like in our home, we convert milk into curd by either adding some inoculum into it or adding some kind of acidic product into acidic sample to it, which converts this milk into curd, and we'll be keeping it outside for the growth of the organism as an incubation period. So these microorganisms such as lactobacillus and others commonly called the lactic acid bacteria lab grow in milk and convert it into curd. So it is a bacteria which convert milk into curd. What I mentioned about inoculum refers to these organisms actually. Okay. So during the growth, the lab produces acid. So what is happening? Why milk is converted to curd? Because of the production of acid, which coagulate and partially digest the milk protein. So what is happening in case of curd? The coagulation of proteins are happening. The proteins get agglutinated, or we can say they get coagulated. They get just float, like they will be like aggregating the proteins present in the milk. And it also improves its nutritional quality by increasing vitamin B12. So in curd, we have more nutritional quality compared to milk, not like in the sense like in milk, you'll be lacking some kind of nutrients, whereas in milk, in case of curd, you'll be having more nutrients. So it improves the nutritional quality by increasing vitamin B12. In our stomach too, the lab play a very beneficial role in checking disease causing microbes. So that is what I mentioned about the normal flora of microorganism in our body. Now, yeah, I mean like as I told, like if you are sitting in your classroom or if you are sitting in your room right now, you could see that some kind of spills might be there, some kind of food material which have like you have left out yesterday, or it will be like some dust particle. There will be like a minute quantity of bacteria. If you have like something unclean or some kind of yeah, untidy areas, or even the water bottles what you use, if they are having some moisture around, like if it is not washed properly also, they'll be having some kind of bacteria inside it. And in our home, we use these microorganisms as well for our welfare. For example, is the dove. In our kitchen, we use these organisms like yeast. You might have heard about that yeast. There is Saccharomyces cerevisiae, baker's yeast. It is called as baker's yeast, which is used for the fermentation. So the dough which is used for bread is fermented by the Saccharomyces cerevisiae. So what they do is that ethanol fermentation is carried out. Ethanol fermentation. So what happens is the sugar molecule present in the carbohydrate, present in the food will be converted into ethanol, which gives the particular odor or the taste or whatever peculiarity, whatever the food is having after that, the fermentation is because of the production of the byproducts and plus carbon dioxide. Okay. Now, another example is toddy. It is a kind of an alcoholic beverage, a traditional drink of some parts of southern India made by fermenting sap from palms. So, then another example is the large holes in the Swiss cheese are produced of a large amount of carbon dioxide by a bacterium named Propionibacterium shermani. So, actually, cheese is also a product of fermentation. Now moving to next part, the microbes in industrial products. So the production on an industrial scale requires growing microbes in very large vessels called fermenters. So, we already discussed about the fermentation process where the carbohydrate is converted into carbon dioxide as well as some kind of like aldehyde or ethanol, like some kind of organic molecule is produced there. Okay, so first we are talking about the yeast. As I mentioned, the Saccharomyces cerevisiae is used for bread making and commonly called as brewer's yeast. And it is used for fermenting malted cereals and fruit juices to produce ethanol. In all the beverages, we use this for process like wine, beer are produced without distillation, where whiskey, brandy, rum are produced by distillation of the fermented brew. So industrially, for like both alcoholic beverages and non-alcoholic beverages use this particular process, that is fermentation. And for the process of fermentation, we need the microorganisms. So Which is the organism we use there? It is a yeast, that is fermented beverages. Now, moving to the antibiotics, which was an pivotal discovery in the medical field or the health industry, which changed many people's life. That means like which changed the whole future of the world, because we could conquer on the effects of certain pathogenic microorganisms. So antibiotics were like the scientist Alexander Fleming, he was working on staphylococcus bacteria, and he observed that a mold growing in one of his unwashed culture plates around which staphylococci could not grow. So that means here you can see this is a penicillium colony, that is a fungi, and around that particular area he saw that there is a zone. That is a zone where no bacteria was found. That means he find out that there is something produced by this penicillium around the organism which inhibited the growth of bacteria. So he found out that it was due to a chemical produced by the mold, and he named it as penicillin because it was produced by the Penicillium notatum organism. On basis of that, he named that particular chemical as penicillin. And later, Ernest Chain and Howard Florey made its full potential effective antibiotic. So penicillin was the first antibiotic to be discovered, and it was a chance discovery. Accidentally, actually, he find it out that antibiotics are the molecules or substances produced by the organism which act against other biological organisms. Okay. So here the definition is, are chemical substances which are produced by some microbes and can kill or retard the growth of other disease causing microbes, that is other pathogens. Okay. Now moving to next part, that is the chemicals, enzymes and other bioactive molecules. So here we have first is Aspergillus niger, which is a fungus. So Aspergillus niger, yeah, produces an acid molecule, acid compound. It is a citric acid. So citric acid producers include Aspergillus niger, which is a fungus, and another one is Acetobacter aceti, which is a bacterium, and it produces acetic acid. And another one is Clostridium butylicum, which is a bacterium which produces butyric acid. And then lactobacillus, just we discussed right now, it is a bacteria present in a curd, and it is called the lactic, the acid produced is lactic acid. Saccharomyces cerevisiae, that is a yeast which produces ethanol by fermentation. So these are the examples of acid fermentation where they fermentation where they produces acid as a byproduct, whereas this is an example for alcoholic fermentation, alcohol fermentation. And along with that, it produces carbon dioxide also. So as we discussed, like yeast is very important microorganism in different industries because it is used for the commercial production of ethanol, and so many microbes are used for the production of enzymes as well. For example, lipase. Lipase is the enzyme which cleave lipid biomolecules. So lipid can be cleaved into glycerol and fatty acids, and this is done by lipase enzyme. And this lipase enzymes are used in laundry detergents, these enzymes are also produced by the microorganisms. So they can be used to cleave the dirt or the fat kind of dirt or dust particle present in the cloth. Okay. And also, like as I told you, they're helping removing all the oily stains from the laundry. So you must have noticed that the bottled fruit juices brought from the market are all clearer as compared to those made at home. So how is it? It is because of the enzymes, that is pectinase and protease. So Protease and pectinase are the enzymes which cleave protein molecules, the enzyme which hydrolyze protein molecules, and they are used in bottle juices. So these, they are more clearer because they can cleave the protein molecules, and they are like clear, clarified because of these enzymes. Another enzyme is fructokinase, which is produced or extracted from the or synthesized by the Streptococcus bacterium, and it is used as a clot buster. Clot buster, what is clot buster? So we know that our body has a normal immunological reaction, that is blood clotting. That is to stop the blood flow when you are having an injury, cut, and as a result they'll be forming a clot and prevent the entry of microbes, prevent the entry of microbes into the body. Okay, this is a barrier, acting as a preventive barrier actually. So what happens is that the streptokinase, which is produced by the bacterium Streptococcus, uh, what they will do is that they will be like, after modifying by genetic engineering, they will be removing the clots. They will be like breaking the clots from the blood vessels of the patients who have undergone myocardial infarctions leading to heart attack. That means, though this is helpful in some people, like who are having, I mean, like the patients who are having blood clots or blood blocks, unnecessarily they are having blood clot, it could be harmful. So what this particular enzyme does is that they will break this clot formation, which is present in the patients who have undergone, like who are like usually the myocardial infarction people will be having a blood clot in between the blood vessels, and by this injection it could be removed. Okay, now moving to the next one is the cyclosporin A. So that is which is used as an immunosuppressive agent in organ transplant patients, and this is produced by Trichoderma polysporum fungi. Remember all these organisms and the product they produce. So here it is acts as an immunosuppressor. So immuno, we know the term immuno refers to a process which is providing a self-protection, that is acting as a barrier to a foreign body or a foreign organism. So suppressing the immune means, if your body, like during organ transplantation, one organ is transplanted from one to another person. So from A person to B person, the organ is being transplanted. So it is so complicated, sophisticated process that they should be compatible each other. Their blood groups, their everything should be compatible. The person who is receiving the organ should be healthy, as well as not sorry. The person who is receiving the organ should accept that organ. If he is rejecting it, if the body is immune, like I mean, like if the body rejects that particular organ, he won't be able to I mean, like withstand that situation. He can't continue his life. For example, if they are receiving a kidney, the kidney should be compatible. The person who is donating the kidney and the person who is receiving it should be compatible each other. The blood groups, all the blood tests should be similar, and it should not be, the kidney should not be acting like an antigen. So there is a need for a suppress the immune system, because our body has a tendency to provide the immune reaction when something is entering our body as like foreign substance. So what is that? Here what we'll use the cyclosporin A, which is acting as an immunosuppressive agent. Next is statins. So statins are produced by the yeast, that is Monascus purpureus, and they have been commercialized as blood cholesterol lowering agents. So blood cholesterol lowering agents mean, we know that our normal blood cholesterol level is 150 to 200 milligram per dl, and if it is higher than this particular level, we will say it is hypercholesterolemia. And the drug what we take for this particular situation or some condition is statins. And we can extract statins from the Monascus purpureus yeast. Okay, so here you have the diagram of Monascus purpureus yeast. So it acts as a acts by completely inhibiting the enzyme which is responsible for the synthesis of cholesterol. So actually, that is HMG-CoA is the enzyme which actually synthesizes HMG-CoA reductase is the enzyme which synthesizes cholesterol. And what the statin do is that it inhibit the enzyme. Now moving to the next topic, that is microbes in sewage treatment. So we know that there are large quantities of wastewater which are generated every day in cities and towns. And major component of this wastewater is human excreta. So this municipal wastewater is also called sewage. So what does sewage means? It is a wastewater which consists of all kind of waste materials which also includes a human excreta. So the treatment of wastewater is done by the heterotrophic microbes naturally present in the sewage. Okay, so here you have the two stages of sewage treatment, that is primary treatment and secondary treatment. Primary treatment is the physical treatment and secondary treatment is the biological treatment. So like if you are having, even in your home also, if you are having some kind of wastewater or like some kind of area which is done with some waste materials, what you will do, you will physical treatment will be done first. We will be taking off the larger molecule which you can separate from the dust. Like if it is a watery body or something like that, you will be removing the physical things or the mechanical or the molecules which are visible to your eyes and you can remove them. Okay, that is the first part, physical treatment. After that, you'll be going and removing secondary treatment will be done as a biological treatment is done. So we know that as this wastewater contains large amount of organic matter and microbes and many of them can be pathogenic. So we have to do the primary as well as secondary treatment. Coming to primary treatment, this involves a physical removal of particles which is large and small from the sewage through filtration and what it is? Filtration and anyone can say sedimentation. Okay, so these are removed in stages, like as I mentioned, physical removal of particles from sewage by filtration and sedimentation. Now coming to secondary treatment, this consists of aeration, activated sludge and anaerobic sludge digester. So there is a stages of secondary treatment. So moving to primary treatment, it involves a primary removal of particles from the sewage through filtration and sedimentation. Now, what is the sections, like each parts or a sequence of this process? First is a sequential filtration, that is to remove the floating debris. If in the water body, if you are able to find out something is floating over the liquid like the water, you could remove it. That is a sequential filtration. You will filter it. Okay. Now next is sedimentation, to remove the grit, that is soil and small pebbles and all. So all solids that settle form the primary sludge. So if in water, we know that if you are like, this is a water body and something is floating over the water body, then you can remove all that things. That is a sequential filtration. Now, if you're finding something is settling down, like soil and small, like that kind of things, you have to like do, again, you have to do the secondary treatment parts, not secondary, in the primary treatment itself. You have to remove that, the soil, the mixed soil and all, by filtration and all, you can do it. And sedimentation also will, like if you're keeping this particular for example, if a beaker is full of water and mix it with soil and all, if you're keeping for sedimentation, the soil will settle down after a period of time. And yeah, after that you could remove it, completely filter it out. So all the solids that settle from the from the primary sludge and the supernatant forms the effluent. Okay, so all the solids that settle form the primary sludge and supernatant forms the effluent. Okay, so this is the primary sludge and this is the effluent. Now the effluent from the primary settling tank is taken for the secondary treatment. Now here we will taking the supernatant part. The effluent is taken for the secondary treatment. Now the primary effluent is passed into a large aeration tank. So now we are moving to the secondary treatment part where it starts with the aeration tank, and this allows vigorous growth of aerobic microbes into flow. So here it is in this aeration tank, oxygen is available, the organisms will be able to grow, they consume the major part of organic matter in the effluent. That means if there are any kind of organic materials, they will be utilizing those organic matter as a source of energy, nutrition, and this significantly reduces the BOD, that is biochemical oxygen demand of the effluent, because the organisms are consuming all the organic materials, and as a result what is BOD? BOD is a measure of the organic matter present in the water. So if the organic matter, for example carbohydrate or any kind of proteins, are decreasing, that means the BOD is also decreasing. So greater the BOD of wastewater, more is its polluting potential. So you have to remember this thing. If BOD is high, that means it is more polluting. Pollution is directionally proportional to the value of BOD, that is biochemical oxygen demand. Now, once the BOD of sewage water is reduced significantly, the effluent is then passed into a settling tank. And here the bacterial flocks are allowed to sediment. And this sediment is called the activated sludge. Okay. So when the BOD of the sewage of water is reduced significantly, so because as the organic matter is reducing, BOD is reducing, and then this effluent will be taken to a settling tank. And here what happens is that the bacterial flocks, the organisms are allowed to sediment. They will settle down. And this sediment is called activated sludge. Actually, we use this activated sludge later. Now, first let's complete this process here. Now, a small part of this sludge is pumped back into the aeration tank to serve as inoculum. Yeah, here we are already mentioning it is acting as an inoculum. That is as I mentioned, that the milk is converted to curd by adding some inoculum. That means a starter organism which is acting as an initiator. Okay, so we can use this in the aeration tank which is acting as an inoculum. Then the remaining part, the remaining major part of the sludge is pumped into the larger tank called the anaerobic sludge director. So we had the primary treatment. After that, it went to the secondary treatment part, whereas the aeration zone has been done. That is air, liquid, water, everything together. The organisms consume the organic matter. And what happened? We have the primary sludge as well as the secondary sludge. Now, as I mentioned, the sludge is pumped back to the aeration tank as well for acting as an inoculum. And then here in anaerobic sludge digester, what happens is that the bacteria will be producing a mixture of gases, such as, have to remain these gases, name of the gases, methane, hydrogen H2S, hydrogen sulfide, carbon dioxide, etc. And these gases form the biogases. So the effluent from the secondary treatment plant is generally released in the natural water bodies like rivers, streams, etc. Now, what happens to the activated sludge? So this activated sludge, like a small part of the activated sludge, is pumped back into the aeration tank to serve as an inoculum. So here it is again pumped back to aeration tank, which is acting as an inoculum in this, as a starter organism. So here what happens is that the organisms will be consuming the carbohydrates, these organic molecules, and reduce the BOD, isn't it? So they will be reducing the BOD, and the remaining part, major part of the sludge is pumped into the larger tank called the anaerobic sludge digester. So during this digestion, the bacteria produces a mixture of gases such as methane, hydrogen sulfide, and carbon dioxide. You have to remember these gases because it is so important. You can expect questions in the NEET exams. There has been like questions regarding this topic as well. Now, so they releases the gases. Now the effluent from the secondary treatment plant is generally released into the natural water bodies like rivers and streams. Okay. So microbes do play a major role in treating millions of gallons of wastewater every day across the globe. Till date, no man-made technology has been able to rival the microbial treatment of sewage. The Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save these major rivers of our country from pollution. So just remember these two plans. And under these plans, it is proposed to build a large number of sewage treatment plants so that only treated sewage may be discharged in the rivers. Now, we'll move to the continuation of the part, that is the microbes in the production of biogas. So biogas is a mixture of gases produced by the microbial activity and which may be used as fuel. So what were the gases? Methane, hydrogen sulfide, carbon dioxide. Now, certain bacteria which grow anaerobically on cellulose material produce large amounts of methane along with carbon dioxide and hydrogen sulfide. So these bacteria are collectively called methanogens or methanobacterium. So the bacterium which synthesize methane is called methanobacterium or methanogens. And the bacteria are also present in the rumen of cattle. Which bacteria? The methanogenic, the methanogens or the methanobacterium. Now, a lot of cellulosic material present in the food of cattle is also present in the rumen, because we know that cattle feed on grasses which are rich in cellulose, and that means the cellulosic material present in the rumen also consists of the methanogens, and in the rumen, the bacteria help in the breakdown of cellulose and play an important role in the nutrition of cattle. And the excreta, that is the dung of the cattle, commonly called the gobar, is rich in these bacteria. So dung can be used for the generation of biogas, commonly called the gobar gas. Understood? So from the grass, that is grass is having cellulose, and this enters the gut or the rumen, and what happens there? It is digested and the methanogens will be releasing methane gas from this process. As a result, the dung will be having the particular bacteria. Now, moving to the next area, that is the microbes in the production of biogas. So this is actually a continuation. Okay. So biogas is a mixture of gases containing predominantly what? Methane. So biogas is mainly rich in which gas? That is methane. And this is produced by the microbial activity and which may be used as fuel. So it is used as a fuel. Now, you know that the microbes produce different type of gaseous end product during growth and metabolism, and the type of gas produced depend upon the microbes and the organic substrates they utilize. For example, in fermentation of dough, cheese, and the production of beverages, the main gas produced was carbon dioxide. Because of what? Ethanol fermentation. Isn't it? And in certain bacteria which grow anaerobically on cellulose material, they produce large amount of methane along with carbon dioxide and also hydrogen. So these bacteria are collectively called methanogens or methanobacteria. Clear? Now, so these bacteria are also present in the rumen of cattle, because we know that the cattle depend on the grass which are rich in cellulose. So a lot of cellulosic material present in the food of cattle is also present in the rumen. So in rumen, the bacteria help in the breakdown of cellulose and play an important role in the nutrition of cattle. And the excreta, that is the dung of the cattle, which is called the gobar, is rich in these bacteria. So dung can be used for the generation of biogas, which is commonly called the gobar gas. Now, Moving to the biogas plant next. So the dung, which is like the dung is converted. We can use this organic for as we discussed right now. The dung is can be used to produce biogas. In the same way, so gobar gas. In the same way, we can use different kind of as we discussed, like every different types of products, like the organic products will be producing different kind of byproducts, isn't it? As I mentioned, in case of fermentation of dark and cheese, like carbon dioxide is released. So the type of gas produced depend upon the microbes and the organic substrate. So here, if you are using like organic materials, waste are used from the animal husbandry. So the crop harvesting, industrial products, processing waste products are used. We can produce what biogas from the substrates. So the technology of biogas production was developed in India mainly due to the efforts of Indian Agricultural Research Institute IARI and Khadi and Village Industries Commission KVIC. So this biogas consists of a concrete tank in which the bio waste are collected and slurry of dung is fed. So actually this is a 10 to 15 feet deep in which the bio waste are collected and a slurry of dung is fed. So here you can see that there is a floating cover which is placed over the slurry. So yeah, let me take that. Now moving to biogas plant. So this biogas plant consists of, as I mentioned, they're consisting of 10 to 15 feet deep concrete tank where the bio waste are collected and a slurry of dung is fed. So a floating cover is placed over the slurry which keeps on rising as the gas is produced in the tank due to microbial activity. So the biogas has an outlet which is connected to a pipe to supply the biogas to the nearby houses as well. And the spent slurry is removed through another outlet and may be used as fertilizers. So cattle dung is available in large quantities in rural areas where cattle are used for a variety of purposes. So these biogas plants are more often built in rural areas. And the biogas produced is used for cooking and lighting. So here you have the diagram of the biogas plant, and this technology of biogas production was developed in India, as we mentioned, by the efforts of Indian Agricultural Research Institute IARI and Khadi and Village Industries Commission KVIC. So you have to keep it in mind because you can expect questions regarding this. So now moving to the next part, that is microbes as biocontrol agents. So what is biocontrol agents? Biocontrol refers to the use of biological method, that is why the term bio. It is a biological method for controlling the plant diseases and pests. So in this modern society, we actually use chemicals to control these diseases and pests, which can be harmful or toxic to the organisms, which can lead to pollution as well. So this biocontrol agents can reduce the toxicity or pollution caused by these chemicals. Okay, so for example, the ladybird can control aphids, dragonfly can control mosquitoes. Okay, so these are the examples of biocontrol agents. Now, moving to another example, that is trichoderma, which is a fungi. It protects the root system and controls the plant pathogens. Another example is the baculoviruses. It attacks some insects and other arthropods. Now, this is another important one, that is Bacillus thuringiensis, which is used to control the wide range of insects as well. It is, we have the Bt cotton. Now, moving to the next area, that is moving to microbes as biofertilizers. So we know that fertilizers are the chemical substances which increase the nutrient quality of the soil. Now what is biofertilizers? Where biological organisms are acting as a fertilizer. So biofertilizers are the organisms that enrich the nutrient quality of the soil. And the main sources of biofertilizers are bacteria, fungi, and cyanobacteria. Now, moving to bacteria. So you have noticed the nodules on the roots of the leguminous plants formed by the symbiotic association of rhizobium. And these bacteria fix the atmospheric nitrogen into organic forms, which is used by the plant as nutrient. And other bacteria can fix the atmospheric nitrogen while free living soil, for example, the acetobacter and azospirillum, enrich the nitrogen content of the soil. Now, moving to, this is the rhizobium, acetobacter, blue-green algae, azospirillum. Now, moving to fungi. So, fungi are also known for its symbiotic association with plants, for example, mycorrhiza. Many members of the genus, that is Glomus, form mycorrhiza. So the fungal symbiont in these association absorb phosphorus as like phosphate, and they absorb the phosphorus from the soil and passes it to the plant. The plant having such associations show other benefits also, such as resistance to root-borne pathogens, tolerance to salinity and drought. So, and also there is an overall increase in the plant growth and development. So they absorb phosphorus, water, like from the soil for the plant growth. Moving finally to cyanobacteria. So here's the same. Cyanobacteria are the autotroph microbes which are widely distributed in the aquatic and terrestrial environments, many of which can fix the atmospheric nitrogen. The Anabina, nostoc, oscillatoria, etc. So in paddy fields and all, cyanobacteria serve as an important biofertilizer. The blue-green algae also add organic matter to the soil and increase its fertility. So now let's look into a few questions from past paper of NEET exams. And here we have from the topic microbes in industrial products. And the question is from May 2024. Now, the question here, you have two columns. In one column, you have the organisms, and other you have the products, byproducts of the particular organism. Now, first you have Clostridium butylicum. Actually, Clostridium butylicum is the organism which produces butyric acid, which is a key product in the fermentation process. That is an example for acid fermentation. So here, butyric acid we have in the third one, A. Next is Saccharomyces cerevisiae or yeast. So yeast is responsible for producing ethanol through alcoholic fermentation or ethanol fermentation. So B. Now Trichoderma polysporum, they synthesizes cyclosporin, which is an immunosuppressive drug, which is used in organ transplantation. So C. Now Streptococcus species. So they produces the streptokinase enzyme which dissolve the blood clots in medical treatment. So D. So that means the correct answer is option A. Moving to the next question. Methanogens belong to which group? Is it dinoflagellate, slime molds, eubacteria, archaebacteria? So we know that methanogens will be producing methane gas, isn't it? CH4. So these are the group of microorganisms belonging to the domain Archaebacteria because they produce methane as a metabolic byproduct often in anaerobic environment such as in the guts of animals or in swamps. So they are mostly extremophiles which will be coming under the category of Archaebacteria. So option D is the correct answer. Moving to the next question, what gases are produced in anaerobic sludge digesters? We already discussed this. The gases were methane, hydrogen sulfide and carbon dioxide. So the correct answer is option B. Now, anaerobic digestion is a series of processes in which the microorganisms break down the biodegradable material in the absence of oxygen used for industrial or domestic purposes to manage waste and to release energy. And the process of anaerobic digestion produces biogas consisting of methane, carbon dioxide, as well as some other contaminant gases which includes hydrogen sulfide. Moving to next question. Microbe used for biocontrol of pests, that is butterfly caterpillars, which is among this that. The answer is option B, Bacillus thuringiensis. This BT, we know that BT cotton, right? Like in the BT cotton, we have the caterpillars which are acting like a pest. So this BT, Bacillus thuringiensis, is a bacterium that produces toxin which is harmful to certain insect pests, including butterfly caterpillars. And it is used widely in biocontrol as a natural pesticide, targeting the specific pest while being harmless to humans, animals, and beneficial insects. So, moving to the next question. High value BOD, that is biochemical oxygen demand, indicates what? We discussed that if the organic matter is decreasing, that means BOD is also decreasing because the organism feed on the organic matter. So if the BOD is decreasing, that means the pollution is also decreasing. So here it is high value of BOD, that means BOD is high, that means pollution is high. So option A is the correct answer. Moving to the next question from May 2019, you have to select the correct group of biocontrol agents. So here the answer will be option B, trichoderma, baculovirus, bacillus thuringiensis. So biocontrol agents, like they include various natural enemies of pests, including predators, parasites, pathogens, and competitors. And they contribute to pest control through mechanisms such as predation, parasitism, and disease transmission, reducing pest populations and minimizing crop damage. Now, what is the role in the pest control? The biocontrol agents help in managing the pests like nematodes, weeds, insects, and mites by regulating their populations in agricultural ecosystem. So they act as a sustainable alternative to chemical pesticides, reducing the reliance on synthetic chemicals and mitigating environmental risk. So what does it mean? Usually if the chemicals are used, it is harmful to all other organisms. But if we are using biological organisms to pest control, they will control the organism and which will not be harmful to other organisms as well. Now, moving to the next question from May 2019. In one column, you have the organisms and the second column, you have the products produced by the organism. So, first one is lactobacillus. So, lactobacillus, you can connect it, right? From milk to curd formation, we did that, discuss that. So, lactobacillus is used in curd formation. Now, second is Saccharomyces cerevisiae, that is yeast, helps in bread making. That is carrying out ethanol fermentation, which produces ethanol and carbon dioxide, isn't it? Aspergillus niger, it produces citric acid. And Acetobacter aceti is responsible for, the name itself is there, aceti, that is acetic acid. It is vinegar production. So cheese is not matching to any one among them. So here the answer is option B. Now moving to the next question, again from May 2019. This is also related to biocontrol agents. So which of the following can be used as a biocontrol agent in the treatment of plant disease? The answer is option A, trichoderma. So trichoderma is a genus of fungi which is known for its biocontrol properties against plant pathogens. It colonizes soil and root ecosystems where it competes with and antagonizes pathogenic fungi through various mechanisms. And trichoderma produces antibiotics as well as lytic enzymes and volatile organic compounds that inhibit the growth of plant pathogen. Okay. So they are biofungicides, they are decomposers, as well as they are biofertilizers. Moving to next question from May 2019. So this is from the topic like microbes and industrial products. Which one of the following equipment is essentially required for growing microbes on a large scale for industrial production of enzymes? The answer is option D, bioreactor. So enzyme production in larger quantity is carried out by bioreactors. They are specialized equipment which is used for large scale production of enzymes and other biological products. So they provide controlled conditions such as temperature, pH and nutrient supply. And this optimizes the growth and productivity of microorganisms or cells producing the desired enzyme. So the large-scale enzyme production often involves the use of bioreactors to achieve high yields and consistency in the product quality. And also the bioreactors come in various designs including stir tank reactors, airlift reactors and fermenters, and they are also essential for scaling up enzyme production for industrial applications. So this was an additional information regarding bioreactor. Moving to the next question. Conversion of milk to curd improves its nutritional value by increasing the amount of which among the following vitamin? We discussed it in the previous slide. So correct answer is option D, vitamin B12. So vitamin B12 is produced in higher amounts during the fermentation process, which enhances the nutritional profile of curd compared to milk. Now select the mismatch. In the options we have Rhodospirillum mycorrhiza. So Rhodospirillum is a photosynthetic nitrogen fixing bacteria which is not associated with mycorrhiza. And mycorrhiza is a fungal plant root symbiosis, that is symbiotic association between fungi and plant roots. So that means the correct answer is option A, which is a mismatch. Whereas Anabaena is a cyanobacterium that fixes atmospheric nitrogen in symbiosis with plants like Azolla. So it is incorrect answer. Rhizobium, it is a nitrogen fixing bacteria that forms root nodules in the leguminous plants like alfalfa. So this is also incorrect answer because there is match, good match. D is Frankia. So Frankia is an actinobacterium that fixes nitrogen in symbiosis with Alnus. That is alder tree. So this is also an incorrect answer. So moving to the next question. Which of the following in sewage treatment removes the suspended solids? So the answer will be option B, primary treatment, because primary treatment is the first stage of sewage treatment which involves the removal of suspended solids and organic matter through physical process such as screening, sedimentation and flotation. Or we can also include like filtration also here. In this stage, the sewage is passed through large tanks where the solid particles settle out as sludge and effectively removing the suspended solids. So the answer is option B, primary treatment. Moving to the next part, which of the following is correctly matched for the product produced by them? So this is from May 2017 and first we have methanobacterium and the product given here is lactic acid. We know that this is incorrect because methanobacterium, as the name suggests, it produces methane gas during anaerobic digestion, not lactic acid. Lactic acid is produced by which bacterium? Lactobacillus. So option A is incorrect answer. B is Penicillium notatum. So that is an antibiotic and the antibiotic is penicillin, isn't it? So this is also incorrect. Penicillium notatum is known for producing antibiotic, that is penicillin, not acetic acid. Acetic acid is produced by the bacteria Acetobacter acetone. So option B is also incorrect. Moving to option C, Saccharomyces cerevisiae, it is yeast which carry out ethanol fermentation or alcoholic fermentation. They are known as brewer's yeast, which is used in fermentation process, which produces ethanol or alcohol and carbon dioxide. Means it is a correct match. So option C is the correct answer. Option D, Acetobacter aceti and antibiotics. As we mentioned right now, Acetobacter aceti is producing acid, that is acetic acid. It is vinegar, not antibiotics. Antibiotics are produced by the organisms like penicillium, streptomyces, etc. So option D is also incorrect answer. Moving to next question, that is from May 2016. So here you have the microbes in one column, the product in another column, and the applications in another column. First one, we have the Trichoderma polysporum, and the product is cyclosporin given here. That is true. The product is cyclosporin, which is acting as an immunosuppressive drug. This is true. That means option A is incorrect answer. Option B, Monascus purpurus. Yes, it is a fungus. We know that. And they produces a statin, which is lowering the blood cholesterol. This is also true. So option B is incorrect answer. Option C, Streptococcus. So Streptococcus produces streptokinase. This is correct. They remove the clot from blood vessels. This is also true. So option C is incorrect. Now moving to Clostridium butylicum. So this is actually known for producing butyric acid, not lipase enzyme, which break down the lipid, that is removal of oil stain, which is used in the detergents and all. So Clostridium butylicum is known for producing butyric acid. So that means option D is the correct answer. Okay. Moving to the next question. The primitive prokaryotes responsible for the production of biogas from dung of ruminant animals include what? The correct answer is simple. Option C, methanogens, which are archaebacteria that produce methane, commonly used in biogas production from organic waste. Now, moving to next question. That is, you have been given two statements here and you have to find out whether they are true or false. First one, antibiotics are chemicals produced by microbes that kill other microbes. This is true, isn't it? Then antibodies are chemicals formed in body that eliminate microbes. This is also true, though this is from the previous chapter, that is from the diseases chapter. Yes, this is true. So both statements are correct. So option C is the correct answer. Moving to the next question, plasmid PBR322 has PSTL restriction enzyme site within the gene ampicillin resistant that confers ampicillin resistance. If this enzyme is used for inserting a gene of galactoside production and the recombinant plasmid is inserted in an E. coli strain. So, what will happen then? So we know that PBR322 is a commonly used cloning vector. So when the gene beta galactosidase is inserted, beta gal gene inserted into ampicillin resistant gene, due to ampicillin resistant gene using PST1, the recombinant E. coli loses ampicillin resistant due to insertional inactivation. So the host cell produces beta galactosidase, but it does not have dual ability or novelty. So the transformed cells cannot resist the ampicillin as they have lost the ampicillin resistance and they do not undergo lysis due to insertion of the beta galactosidase gene. So that means it will not be able to confer ampicillin resistance to the host cell. Why? Because it is having the beta galactosidase enzyme, the gene which is releasing the enzyme which it is inserting. By insertion of this beta galactosidase gene, it loses its ampicillin resistance. That means because of the insertional inactivation. So by this question we are coming to an end to this class. So yeah, so this is from the chapter Microbes in Human Welfare.